All right, so today's lecture is going to be about phylogenetic analysis, which tries to put things in evolutionary perspective. So you try to find relationships between entities, which could be either genes or species, and construct the evolutionary history of those entities. So the definition is you construct the line of descent or identify the evolutionary development of any species or gene, or the origin and the evolution of a division, group, or race of animals or plants, or other organisms. So the goal in constructing phylogenetic trees and studying phylogeny is to understand the evolutionary history and assist in epidemiology, such as in infectious diseases. You would want to identify where the infection originated, and you can do that by relating this the samples that you collect with each other to trace back their origin. And also genetic defects. You can look at how genetic defects have evolved or have been inherited through a lineage. It also aids in functional prediction of genes. So if you can identify the relationship of a gene with other genes, you can make some inferences in terms of their function. If things are related through the evolution, you would assume that they share similar functionality. Okay. Phylogeny helps understand microbial evolution, and it also helps understand adaptive immunity. Phylogenetic trees have been constructed from a long time ago, even before we had gene sequences. One of the early examples of this is from 1866 by Ernst Haeckel, and this is mainly based on the anatomy, so how these organisms looked instead of their genes. We didn't have gene sequences then. Okay, so some of these placements are inaccurate, and they were readjusted after we were able to look at the genetic sequences of the organisms and were able to more accurately trace back the lineage of different species. One of these corrections is how human relates to other animals. So in the pre-molecular view, that's before we had the genetic sequences, humans were a separate branch, and these other animals were clustered together. Okay, so this is based on sort of the intuition that an intuitive analysis of how these animals looked like, how their anatomy was, and so on. So humans felt like they were special; they should be placed in a different place. Okay, so it was more political than perhaps biologically grounded. Okay, but after we had the genetic sequences and we were able to compare this genetic material of these species, humans were found to be more closely related to chimpanzees and bonobos than gorillas and orangutans. Okay, so this means that if you look at the genetic sequence of humans and chimpanzees, they are closest to each other than closer to each other than the genetic sequences of chimpanzees and gorillas are. Okay, and we will see how we can construct this tree ourselves from a set of distances. And this tree was generated from mitochondrial and nuclear DNA from DNA hybridizations. So you do not necessarily need to know the entire genetic sequence of these organisms to construct these trees. In this experiment, they used DNA hybridization. So you just take the DNA of two species and see how well they hybridize. Okay. So if things are complementary, if two DNA strands are complementary, they will hybridize together, right? And the level of complementarity will determine how strongly they bind to each other. Okay. So the stronger they bind, the more similar the DNA sequences are. And that level of complementarity can be measured and be used to construct this evolutionary tree. All right. Another example is forensic analysis of HIV sequence data. I can't remember which state this was in, but there was a patient who had HIV, and her only risk factor was that she had a dentist who was also HIV positive. Okay, so I believe the Center of Infectious Diseases made a study where they sampled the HIV sequences from the dentist and some of his patients who had HIV and did not have HIV, and also they also sampled from HIV patients from that local neighborhood, but who were not that dentist patients, and they saw that the dentist's patients clustered together, whereas HIV positive patients not related to the dentist were clustered separately. Okay. And there were some other patients of the dentist, F and D, which probably got their HIV from some other source than the dentist. Okay.
So basically, those people got the HIV from.
We can't really claim that. We just say their HIV sequences or their HIV viruses came from a closer source. Okay. So if you think about the HIV spreading and mutating, you can't really claim that one received from the other. They just have a shared source. The infection is the spread of the infection is closer in that region for those individuals. Okay. All right. Okay. Another example for the use of phylogenetic trees is genetic anthropology, and this is where you can trace the migration events or historical lineage of individuals. And for this, two different types of DNA material can be used or has been used in the past. Mitochondrial DNA helps you trace the maternal lineage, because the mitochondria is only comes from the mother, whereas the Y chromosome helps you trace back the paternal lineage. Okay. All right. So, for instance, if you look at the migrations in history, let's say if predominantly males migrated to a different region, let's say they were soldiers and they went and inhabited a new place, then the paternal lineage, the Y chromosome, would reflect that. Okay. All right. So the reason mitochondrial DNA is useful is that it's transmitted through maternal lineage, and its mutation rate is faster than the nuclear DNA. So that makes it very useful for comparing closely related individuals. Okay. So if you look at the DNA from the nucleus of two closely related individuals, let's say that are third, fourth cousins, their DNA material will be very similar. Okay. But if you look at their mitochondrial DNA, which mutates faster, then the DNA material is less similar. So you can differentiate better if you use something that mutates faster. Okay. On the other side of that is you don't really want something that mutates too fast, because then you may not be able to find similarities and know that two sequences are related to each other. All right, so some large projects on phylogenetic trees and constructing evolutionary history is Genographic project, where it's the largest DNA database for genetic anthropology. It contains 5,000 populations and 100,000 people. HapMap is another project which is more disease-oriented. They construct a haplotype map of the human genome and sort of deposit the common patterns of genetic variation in humans. And HapMap becomes very useful when you are studying diseases and which SNPs would be responsible for a certain disease. The Journey of Mankind tries to uncover the global journey of man over 160,000 years. So by sampling individuals from different locations and from different races, you can find you can figure out how these populations journeyed or migrated through the time. Okay. All right. So let's now get into some theory. When we look at a tree, we can classify them in different ways. One of the ways that we can classify them is based on whether they are rooted or unrooted. So on the left, you see an unrooted tree. Let me open up my... Okay, there we go. On the left, you can see, because my thing is not working. There you go. All right. On the left, this is an unrooted tree. Okay, so there's no root to it. So we don't know which element starts the history. Okay, so it doesn't have any time progress. All right. So everything is sort of in a flat relationship. There's no central node that you can point to. So this node could be the root, that node could be root, and so on. So we don't know which one the root is. There are ways to take an unrooted tree and make it rooted. Okay, and that's usually done by comparing everything with an external node. So let's say you take another bird and compare with everything else and reconstruct this tree and figure out that this one would be clustering with this external node, then this would be your root. Okay? All right. So the unrooted trees only reflect the distance. The branch length would usually reflect how different things are, how different two birds are, or how distant their genetic sequences are. You don't really need to have genetic sequences. You can define the distance in some other ways. Okay. Let's say you define the distance based on how what the difference is between their average weight of the two species of birds. Okay. So the distance measure can be arbitrary, in a way. Okay. The genetic sequences gives you more accurate evolutionary history. Okay. When you have a root, you also have a direction of the evolution. So when you have a root, the evolution progresses this way. All right. So this is the earliest time, and the leaf nodes are the most recent species. The internal nodes, we may or may not have them. So we may have fossil samples where we can reconstruct the genetic sequences of the internal nodes, but we do not necessarily need those sequences to construct this tree. Okay, so even if we don't have these sequences, we can sort of estimate what they would be based on what we have nowadays. Okay. So usually you have only the leaf samples and not the internal nodes. So the internal nodes will be ambiguous most of the time because you don't really have samples that dates back to 100,000 or millions of years ago. All right, so let's do an exercise here. Let's ignore this branch. If you have a rooted tree with n leaf nodes, so let's say n taxa. So those are the samples that we have. We have n birds. Can you tell me how many branches I'll have in my tree in terms of n? So log n is the height of tree usually. It's assuming it's a binary tree. Log 2 of n. So how many branches would I have if I have n samples in the leaves, n leaf nodes? N minus 3. Okay. Any other answers? I'm ignoring this branch, okay? N minus 2? Somebody say that? Okay. So I guess the best way to solve this problem is to start small. If you have only a single, let's not consider the single case. Let's say you have two birds. That means you have 2 branches. Okay. If you have 3 birds, you're adding 2 more branches, right? I guess having that extra branch is convenient, so I'm going to change back and do have that extra branch. Sorry for changing the problem on you. Oh, my computer crashed. It's back. All right, so if you start with 2 birds, you have three branches, right? If you add 1 more bird, you add 1 more branch. If you add 1 more bird, each bird you add adds 2 new branches, but that is not correct because the nodes I'm adding are not leaf nodes. All right, so I'm trying to find the best way to explain this to you. So if you look at this part, you have 4 birds, 1, 2, 3, 4, 5, 6, 6 branches, right? Okay, I'm not going to be able to get out of this. All right, so I'm redefining the problem. Okay, I'm going to define the number of nodes as the number of total birds I have in the graph. All right, that's going to make it an easier problem. Instead of just looking at the leaf nodes, let's look at the number of total birds we have. So if you have 3 birds, you will have 2 branches. Any new bird you add will add another branch. Okay? Any new bird you add gets its own branch to get connected to the tree. So you start with n minus 1 branches, where n is the number of birds, and every new bird you add adds its own branch. So n minus 1 doesn't change. Okay. So if n is the number of birds you have in that entire tree, n minus 1 will be the number of branches you have. All right. Okay. All right. Somebody said that the height of the tree would be log n. Okay. So what does that mean? If you have, let me give you an example of a binary tree. So a binary tree is where at each node the branching factor is 2. So each node will have at most will have exactly 2 children or no children. Okay. So that's a binary tree. Sometimes, let's say when you're looking at a family tree, nodes may have more than 2, so that's not a binary tree. Okay, so families can have more than 2 children, which makes them non-binary. So let me draw an example of a binary tree. Oh. Board. I have to make sure it's not permanent. Okay. So a binary tree is where each node will split into 2. Okay. Let's count the number of nodes we have based on the height of the tree. Okay. So if you have height equals 0, you will have only a single node. 1 node. At level 1, so let's say this is the first level, you will have 2. At each level, you multiply the previous level with 2, right? Because each of the previous nodes will have 2 children at the next level. So this will be 4. And at the next level, you will have 8. Okay. So this is level 0, level 1, 2, 3. If you look at level 4, you'll have 16, right? So each level just multiplies the previous level by 2 because each node has 2 children. Now this is just 2 to the power 3, 2 to the power 2, 2 to the power 1, 2 to the power 0. If I tell you that I have a tree where I have n leaf nodes, you should be able to figure out how high the tree is. And you will just say 2 to the x equals n. Take the log of both sides, so x becomes equal to log 2 of n. So for a tree that has n leaf nodes, the height of, if it's a binary tree, the height will be log 2 of n. All right? Okay. And this will, I'll tell you where this comes into play when we are looking at the computational complexity of constructing phylogenetic trees. All right. There are some different types of rooted trees. So one of these is shown in A, cladogram, where the branch lengths are irrelevant. Okay. The additive tree is where branch lengths reflect the evolutionary divergence. So the evolutionary distance between two species is reflected by the length of the branches. So in this example, for instance, these two birds are 9 units apart. Let's say they are 9,000 years apart. Okay. So you would usually be told what this unit reflects. All right. And you have to add up both branches to get the distance between those two birds. If you are looking at the distance between this internal node and the leaf node, then it would be 3. Okay. All right. So additive trees give relative measure of evolutionary divergence, but it doesn't contain any information on time. So it does not contain any information on how many years are between two branches. It's just a unit. Okay. So everything is relative within the tree. An ultrametric tree is an additive tree where you have an idea of how much time each branch reflects. So the branch lengths now have a corresponding time unit to them. So to get from the ultrametric tree to from an additive tree to an ultrametric tree, we need to have a notion of evolutionary time. And to have that, we usually assume a molecular clock where a constant mutation rate is assumed. Okay. So what does that mean? We can, for example, assume that we have 1 mutation per 1,000 years. Okay. Then based on that assumption, we can convert the mutation distance to a time distance. Okay. All right. So in this example, going from this figure C to figure D, we are creating a rooted tree from an unrooted tree, as I told you in a few slides back. You can use an external node. All right, so the external node should not be related to any of the other birds or any of the other species. because otherwise it would, for instance, attach itself somewhere in between. So you don't want that to happen. So you will just add a new species which you know is different from everything else, and that will help root that tree. Okay, so you'll find where things started by adding a new external species that would help relate everything else. Okay. All right. So how do we construct phylogenetic trees? If we have the genetic material, we can calculate the distances or scores. Let's say using dynamic programming, you find the sequence alignment scores, or let's say we use the percent identity, okay, which is an easier measure to think about. The percent identity measure tells you what percentage of the aligned residues are identical. So that's a similarity score, right? Because the higher it is, the more similar things are. When we are constructing phylogenetic trees, it's easier to talk about distances instead of similarities. So to convert a similarity score to a distance score, you just take the negative or subtract it from a maximum number. All right. So for the percent identity case, instead of using the percent identity, we'll just say 1 minus percent identity. So the fraction of residues that are not identical will give us the distance. All right. So in this example, we are given a matrix where these numbers are generated in one way or another. So let's not worry about how they were generated. They were, let's say, generated by sequence comparisons. All right. But they could very well be constructed from any other method. But once you have this distance matrix, you would like to go from here to this tree. Okay. So the UPGMA method, the unweighted pair group method with arithmetic mean, is a step-by-step iterative algorithm where you find the closest pair of groups, so the closest pair of species from this matrix, and then you cluster them into a single group. And you find the next closest pair, and the closest pairs could be between two groups, a group and a species, or two species. Okay? And then you pair them up, you cluster them together, that gives you a new group. And you repeat this step until you have a single group left. All right? And as you cluster things, you have to go back to this distance matrix and modify it. Okay? So I'm going to take a break from this presentation and go into a different one here and tell you more about clustering. So clustering can be used as a data visualization method when you are doing data analysis. And it also, in evolutionary studies, it tells you the phylogenetic relationship between entities. This is closely related to classification problems, but in classification, we do supervised learning where we have a sample and we try to find a label for it. Okay, so a sample, let's say, is a gene sequence, and a label, let's say, is where that gene would go in the cell. Does it go to the cytoplasm? Does it go to the mitochondria? But the localization label will be a class that we would like to identify. So that's a classification problem. All right. Whereas clustering is unsupervised learning, meaning we take the data as is and we try to organize it without any label in mind. Okay. All right. So in unsupervised learning, we find natural groupings of instances from unlabeled data. So unlabeled means we don't have classes associated with different entities, or even if we do, we ignore those labels when we are doing clustering. Okay. All right. So the goals of clustering are data reduction, finding different data types or different data classes, and also they could be used for outlier detection. Okay, so if you cluster things together, create a phylogenetic tree, and find that a sample is clustered separately from everything else, then that would give you an outlier. Okay. There are different methods to create clustering, to obtain clusterings. And there are also some measures that tell you how good a clustering is. And those measures are usually based on two things: the intra-cluster distances, which are the distances between entities within a group, okay? And the inter-cluster distances, which are the distances between groups of objects, so groups of between pairs of clusters, all right? So ideally, you would want to minimize the intra-cluster distances. You want things that are closer to each other to be in a single cluster. And you would want the inter-cluster distances to be maximized. You want to push away things that are not clustered together. Okay? All right, let me skip that. Okay, so these clusterings can will need a distance measure to work. So you need to be able to specify what the distance is between two entities in order to create a clustering of them. What are these? What are the options for distance measures? The Euclidean distance is probably the most commonly used one. My computer keeps crashing. Okay. But there are also some other options, such as Manhattan city block, which is computationally less complex, and also weighted Euclidean, where each axis is weighted. Okay, so if you have different ways to measure how two things are similar or how two things are distant, let's say you have the mitochondrial DNA, and then you have the ribosomal DNA. Okay, so you can either just take the average of two distances, or if you think one type of information is more important, you can give that a higher weight. Okay, so weighted Euclidean is where you can weigh different information differently. Instead of putting everything, taking the average, you add weights based on how important things are related to each other. Okay. So these measures are metric, but there are also some non-metric measures. Non-metric means they don't usually satisfy triangle inequality. So if A and B, if I am similar to somebody and we have a third person similar to that somebody, you would assume that me and that third person are also similar. Okay, so if the distance measure that you are using is a metric, then you can claim this property. But if it's not metric, you can't really claim that the fact that me and a third person are similar to a central somebody, you cannot claim that me and the third person are also similar. Okay, so depending on what distance measure you use, you may be able to make different claims on how things are similar. All right, let's skip that. Okay, so an important decision that you would make when you do clustering is how many clusters do you generate. Okay. So for phylogenetic trees, it's a hierarchical tree. We cluster things all the way to the top. But if you wanted to cluster, let's say, things into flat groups without creating a hierarchy, then you would have to make a decision on how many clusters you want to generate. Okay, so you could make 2 clusters from this data, right? But if you decide to go further down, you could find subclusters within those, right? So things within these 2 clusters that are highly similar to each other. So depending on at what level you look at, you would get different number of clusters. So this in a way relates to phylogenetic trees. In phylogenetic trees, it's a hierarchical tree. We cluster all the way to the top. So the first level would make up the first level, all right? And then within that, we cluster things again into more clusters, subclusters. Okay. All right. Clusterings can be exclusive or overlapping. In exclusive clustering, each data sample belongs to its own class, but in overlapping clustering, a data sample can belong to multiple classes. An extension of that is fuzzy clustering, where the membership is now a floating point number. So a data sample could be 60% a member of cluster A and 40% member of cluster B. So the membership is no longer a binary decision; it's a fuzzy measure. Okay. All right. Let's skip those definitions and go down to the algorithms. Okay, so this is a taxonomy of the clustering methods. For phylogenetic trees, we will create hierarchical, but for some of the other applications, k-means is usually the most commonly used clustering method. Okay, so let's get to hierarchical clustering. In hierarchical clustering, we produce a set of nested clusters, okay? And this can be either visualized as a dendrogram or as a Venn diagram on the right. And when you look at this tree, this dendrogram, any two samples will be grouped together at some level. So let's say this X2 and X3 are grouped at some level. And that level is, there's a scale here. So they are connected at 85% similarity. Okay. And once two samples are grouped, they remain grouped. So the fact that you are grouping with something else doesn't break X2, X3 group. Okay, so once you commit a grouping at a previous level, that grouping stays the same. All right, so there are two ways to construct hierarchical clusterings. One is agglomerative, where you start points as individual clusters. And then you find the two closest pairs, you put them together, and then repeat this process until you have a single cluster left. In divisive method, you start with, you assume everything is in a single group, and then you divide them into two groups, and then you subdivide those into two more groups, and so on. Okay, and you repeat that until you have a single leaf node in each group. And depending on the method you use, you may get different results. All right. My computer likes to think. Okay, it came back. All right. So what do we do here? So we have a distance matrix, okay, given on this figure on the right, and I'm gonna be using a distance matrix and a similarity or proximity matrix interchangeably. You can always go obtain one from the other. Okay. So we have these individual points. Let's say P1, P2, and we have a matrix, distance matrix or similarity matrix that tells us the relationship between these points. And at some intermediate position, at some intermediate step, what we'll have is some of those points will be clustered together, and some will still be left alone. Okay, and we will have a set of trees. Okay, So at any intermediate level, we will have a set of trees, and those trees could be either a single node or a set of nodes. Okay, and at the end, what we will have is a single tree in this set. And as we find the closest two clusters and merge them, we also need to update the distance matrix. Okay. So the distance matrix will reflect that intermediate step. So how do we update the proximity matrix or the distance matrix at an intermediate step? Let's say we have these points and we cluster the second and the fifth one. So we had we want to create this new matrix where everything else is the same except that we need to have new numbers for that row and column. Okay, so if you look at here, we have our original matrix, and we found out that C2 and C5 had the highest similarity or the lowest distance, and we decided to cluster them together. So what we need to do is take this column and this row and use those to create the new matrix. And the way that we will create the new matrix will give us different results. Okay. So how do we define intercluster similarity? So taking two samples or two clusters and merging them, and going back to the matrix and updating the matrix means you are defining a distance from a cluster to the other point or to the other cluster. So how do we define similarity or distance between two clusters? So we already know the distances between these points, right? So that was given in the original matrix. Okay. What we could do is find the minimum distance. Let's say the two closest points between two clusters, and then use that to give us the new distance between those two clusters. Okay. So what do I mean by the two new clusters? So let's say this is C2 and 5, so that's one cluster, and C1 is another cluster, okay, which right now has a single point, a single sample. And we want to identify this inter-cluster similarity. So in this case, we have C2, C5, and the other cluster has only C1. So these C1 to C2 and C1 to C5, we already know these distances from the original matrix. So those are C1 to C2 and C1 to C5. Okay. We need to use those original distances and create that new distance measure. So one definition or one method we could use is to find the minimum distance between the two clusters and use that as the new distance value. The other is the maximum. So look at the two clusters, find the 2 points that are farthest away from each other, and use that distance to define the distance between the two clusters. You could do group average, which is the most commonly used one. You find all pairs of distances between the two clusters, and then you take the average of all those distances. You could find the distance between centroids, where you find the centroid of this group and the centroid of that group, and you calculate the distance between those 2 points. Okay. There are also other methods that you could use to generate that new matrix. Okay, let me skip that. Here, this is a better. All right, so let me go back here. So in this example, what we are doing is clustering these points. We have 6 points, and using different different measures to calculate the inter-cluster similarity. So in the first step, you find the two closest points and cluster them together. So for all definitions, the two closest points are the same. In the next step, you find the two closest methods and you merge them. All right. In the next step, things start getting different now. The way you define the distances between clusters now change which clusters are going to be more similar to each other. So in the first example, where you define distances in terms of the minimum distance, the second and the third points are going to be closest to each other. So that distance is going to be smaller than this distance right here. That's why the minimum method will cluster those points, and the maximum method will cluster these points. Because as far as the maximum method is concerned, the distance between these two clusters is the distance between 5 and 6. So that's larger than 4 and 6. Okay. So for the max definition, the two closest points, or the two closest clusters are 4 and 3 and 6. And you keep doing that to get the final answer. All right. Okay. So the advantages of the main advantage of the hierarchical clustering is that you don't need to decide on any particular number of clusters. If you are doing k-means clustering, you need to pre-specify how many clusters you would like to obtain. But if you are doing hierarchical clustering, you can take your tree and cut it at any level to obtain flat clusters. So what do I mean by flat clusters? Let's say we want to obtain 3 clusters. You can cut the tree at this level and obtain 3 clusters. 1, 2, 3. So any branch that you cut through will give you a new cluster. Okay. A weakness of the hierarchical clustering is that they don't scale well, meaning if you have a large number of samples, you will need the time complexity is approximately n cubed, where n is the number of samples. Okay. So it may not be feasible if you have too many samples in your data. So how can we get the n cubed complexity? At each step, you are looking at the distance matrix, right? So distance matrix will have n square numbers in it. So that's an n square scan. The complexity of looking at the distance matrix is n square. Even though that complexity reduces as you go up, because the size of the distance matrix reduces, let's assume that it's n squared the whole time. All right. And you need to do merging of things n times. So you repeat each step, finding the closest pair and merging them. You do that n times, where each time you do it, it's an n squared operation. So it gives you n times n squared, n cubed total complexity. Okay. Another weakness is that once you cluster two things together, the decision cannot be undone. Okay. What I mean by that is you don't, if you cluster with a new sample, the new clustering doesn't change the previous groupings. Okay, so once you commit, things will stay like that. And this was a problem that we also saw in multiple alignment methods that were based on phylogenetic trees, right? So you would take two sequences or two sets of sequences that were closest to each other, and you would align them. But once you align them, that alignment didn't change on no longer. So you sort of committed yourself to that set of alignments, and you cannot correct that alignment when you add new samples to it. Okay. So here, there's a very similar problem here. All right, let's skip that. Let me quickly show you k-means clustering, even though it's not very related. It's related to hierarchical clustering, but I'm going to show it to you because it's useful to know. Okay, so all right. So in k-means clustering, instead of finding clusters hierarchically, where you have groups and subgroups, in k-means clustering you have flat groups. So you have one group that contains a number of points and another group that contains another number of points, but you do not subgroup those groups. Okay. So when you create subgroups, that means you are obtaining hierarchical trees or hierarchical clustering. So how do I do k-means clustering? If I am given a number of points, you will start off by initializing cluster centers. So I will have these points as cluster centers, and you can initialize them either randomly or just pick any point from your data and assign them to be your cluster centers. And in this example, I'm creating 3 clusters from this set of data points. Okay. So once I've picked random cluster centers, I can look at all the points and see how they are related to these cluster points. And each center will claim some of these data points. How do they claim it? If you take any of these axes, any of your data samples, you find which cluster center they are closest to. This is the closest one. And you assign that this X is a member of the first class. And that membership can also be sort of visualized by drawing a line. Anything above the line is going to be a member of the first class or first cluster. Anything below the line and above the second line are going to be members of the second class, and everything below that are going to be members of the third class. Okay. So once you assign these memberships, you can redefine the cluster centers. All right. What you do is look at all these x's within, let's say, group 3 and find their average. Find their centroid. That will be the new cluster center. So you take this cluster center and then it'll move, let's say, over here. Okay. So this guy moves over here, there, and there. And then you repeat this step. You reassign all the data points to the centers that they are closest to, and redefine the cluster centers. Repeat the steps until the memberships do not change. Okay. So until you redefine the clusters and all the samples remain in the same group. And that will give you k-means clustering, where this is, let's say, the first group, second group, and third group. Okay. Any questions? Okay. Let's go back to the phylogeny tree and see how we construct this tree on the right. So we are looking at this matrix, and we need to find the smallest number. The smallest number is this one here between man and monkey. All right. So the distance between man and monkey is 1. So we link them together, man and monkey. And the height of the level at which they merge is going to be 0.5. It's going to be half of their distance, because their distance is 2 branches. That's going to be 0.5 plus 0.5. All right. So it gives you 1. So you divide the distance by 2 to obtain the level at which they merge. So you can draw this scale of distances. Man and monkey merge at 0.5. So once I merge man and monkey, I need to go back to my matrix and update all the distances. And in this example, I'm using the the average distance definition, where the distance between clusters is defined as the average distance between all pairs. So I will take man and monkey, remove it from the matrix. And I will add a new row to this matrix. Let's call it man and monkey. Okay. So what's the distance between man and monkey, this new group, and turtle? Any answers? So after I merge man and monkey, I need to find these new distances. I need to create a new matrix from after that merging. And to create that new matrix, I need to fill in these numbers. So what's the distance between the man and monkey group and the turtle? 19? Okay, so we're gonna look at our original distances. Okay, so the distance between man and turtle is 19. The distance between monkey and turtle is 18. So that makes it 18.5. Okay. All right. So you just take the average. This row no longer exists. Let's calculate the distance between tuna and man and monkey. The distance between monkey and tuna is 32. The distance between man and tuna is 31. That gives us 31.5 average. Okay. Next is chicken. Chicken and monkey is 17. Man and chicken is 18. That gives us 17.5. Moth and monkey is 35. Moth and man is 36. That gives us 35.5. Okay. And since this matrix is symmetric, only lower half is shown. Okay. That's why we only have one triangle. We don't really need to fill in the entire matrix because it's symmetric. Next is monkey, which no longer exists. Next is dog. The distance between dog and man is 13. The distance between dog and monkey is 12. So it gives us 12.5. All right. So once this operation is complete, we are back to where we started in terms of what we need to do. We have a matrix. We just need to find the smallest element in it. Okay. So what's the smallest number? We took this column out and this row out. The smallest number looks like 8, right? So the smallest number is chicken and turtle. So we need to combine chicken and turtle. Their distance is 8, and in the meantime, let's draw them on our tree. We'll draw this new branch between chicken and turtle, and their height, the height at which they merge is going to be 4. It's going to be 8 divided by 2. 8. Oh, sorry, 4. Okay. And then we repeat the same operation for updating matrix that we did for man and monkey. So combining chicken and turtle, we'll get rid of this row, and we'll get rid of this column. So we are adding a new row and column to this matrix. Okay. So this one doesn't need to be calculated. This one doesn't. What's the distance between tuna and this new turtle and chicken? TC. So you need to find the distance between turkey and tuna and chicken and tuna and take their average. Okay. Turtle and tuna was 27. Chicken and tuna is, turtle and tuna is 27. And chicken and tuna is 26. So that gives you 26.5. Okay. And you repeat, you do it for all entries, fill up the matrix, and then you are back to where you started in terms of what you need to do, and repeat this process until your entire matrix contains just a single row.
So the distance between the man and the monkey is 1. The total distance, right, is 1?
Yeah.
And then So why is it 18.5? Because the monkey and the turtle is 18, but the man and the turtle is 19? And then the man and the monkey of one between them? So that's like half?
Which?
The first piece, yeah.
First column?
Mm-hmm.
Where should I point my finger at?
The man and monkey.
Man and monkey versus turtle?
Yeah.
Okay, so how did I find man and monkey versus turtle? I looked at man and turtle and monkey and turtle and took their average. Man and turtle is 19, monkey and turtle is 18. Their average gives me 18.5.
Okay, so you don't care about the distance between the man and the monkey anymore?
Yeah, so once you merge man and monkey at distance 1, you don't really care about that distance anymore. The new matrix will not really make use of that number 1. Okay. All right. So we're good? Yes.
How would you compare man monkey to turtle?
So how do I compare two groups? Okay. So as far as the matrix you have, man and monkey is just going to be a single entry. Chicken and turtle are just going to be a single entry. So when you perform the merging operation and update your matrix, you will have a new matrix where man and monkey is just going to be one row. So it's just a new name. Okay, you have a matrix. It's just that things have more complicated names, but they are still single entities. Okay, so you don't do anything differently. You don't look at your very original matrix anymore. You work on the new matrix that you have. Okay. All right. So when you continue merging things like that, you get this entire tree. Okay. And this tree is labeled primates, birds, reptile, mammal, reptile, fish, and these labels are sort of knowledge based. They are not part of the they're not part of the data. Okay, so it's an expert label given to the tree. Okay, let's take a quick break before we move on. All right. So given a tree, we can cut it into different portions. Remember when I told you that you can cut the tree at any level to give you a different number of clusters. Okay. So in this case, we'll cut it to give us just 2 clusters, but we won't cut it at a level. We'll just cut it at a branch. So when I say cutting it at a level, is to take a knife and just slice it like that. That means you're cutting it at a certain level in the evolutionary history that will give you as many clusters as the number of intersection points. So you get 3 clusters, right? But if you cut it at a branch, you're going to get 2 clusters. One of them is going to be the small one, and the other one is going to be everything else. Okay? So depending on where you cut, you will get this sort of matrix. If you cut it at this branch, sea lion and seal are going to be in one group, and everything else is going to be in another group. So one group is shown as X's in this matrix, and everything else is shown without an X. If you cut it at this branch right here, you will get raccoon and bear in one group, and everything else in another group. Okay? If you cut it over here, you get raccoon, bear, and dog, and everything else. All right? If you cut it at this branch, you get raccoon, bear, dog, sea lion, and seal. If you do it over here, you get also the weasel. Okay? So you can repeat this cut at any branch to get this matrix. I mean this split format. And these splits are useful for evaluating how 2 different trees are similar to each other. Okay? let's say you generated a tree from a mitochondrial DNA, you got one tree, and then you generated a new tree from ribosomal DNA. Okay. Or you took the pre-molecular view of the species tree for humans and chimpanzees and other animals, and you took the one that you have after the pre-molecular view, and you wanted to see how they compare to each other. Trees are difficult to work with, so you need a more simplified representation to compare them. So one way to compare two trees is to count the number of times that you have the same split. Okay. So if you have two trees, let's say this is one and this is the other one, you will generate these different splits for each tree. Okay. And so on. And you will see how many times you have the same split occurring in the same between the two trees. And those number of times that they share the same split is going to tell you how much those two trees agree with each other. All right. The reason we need to have a way to evaluate this similarity or the agreement between the different trees is that there are a lot of methods to generate trees and a lot of data, different types of data to generate different trees. So we need to sort of find a new tree that every method or all the data agrees on. Okay. Another format to show a tree is this parenthesized format, and this is the one that I'm going to use in the programming example, where you take the closest pair, let's say raccoon and bear, and you parenthesize it. Okay, you separate it with comma and you put it in a parenthesis, and then you take the next class that is merged, and you wrap it with parentheses. So raccoon, bear is one group, and dog is clustered with them. And then sea lion is seal is another group. Okay, and that is grouped with the previous raccoon, bear, dog group. All right, and so on. Okay. Here in this representation, I'm using different parentheses like curly braces and square brackets and parentheses, but that's really just for visual purposes. If you had just regular parentheses for all of those cases, it still represents the same information. Okay. All right. Another way to sort of evaluate a tree is by bootstrap analysis. If you have a large data set from which you can generate a tree, what you can do is take a portion of the data set, generate your tree. Okay, let's say you take 90% of the sequences, generate your tree. Or let's say if you are looking at 100 genes across different organisms, you can say use 90 sequences to generate a tree. Okay, and you can randomly pick which 90 you are using, and you can repeat this many times. Each time you do that, you will get a different tree. Okay, or you may get a different tree. Bootstrap analysis is exactly that. You take your data, you randomly subsample it, you randomly pick a smaller portion from your data set, you get your result, and you repeat this process, and you evaluate how many times a clustering of species or a subgroup is present in all the trees. Okay? All right. So what this figure shows is a tree with bootstrap numbers on it. So that those numbers tell you how much that branch is supported by the data. 50% of the time, out of all the random trees that you generated, 50% of the time, raccoon and bear branch was here. Okay. and you can evaluate that how many times things happen by looking at their split matrix. 50% of the time you had this branch. Okay. 80% of the time these things clustered together. 100% of the time C line and seal were merged together, were clustered together. Okay. So once you have these, you can prune out the branches or the clusterings that are not highly supported from the data. Okay, so that means if they're not supported by the data that much, they may be there just by chance. Okay, or it may mean that we do not have sufficient information, sufficient data to claim that that clustering is real. In figure B, what I'm doing is I'm taking my bootstrap tree and applying a threshold of 60%. Okay. So if anything has support less than 60%, I will assume that I cannot really have that clustering. I don't have enough support for that clustering. Okay, so this clustering was supported 50% of the time. It doesn't pass my 60% threshold. That's why I'm going to do a flat clustering of this raccoon, bear, and dog. Okay. I don't have enough information to subcluster raccoon and bear out of dog. So this will be as one group. Okay. Again, that still has 50%. I need to go one higher level to 75%. Okay. So dog, bear, and raccoon will now be at this level where I have 75% support. Sea lion and seal are 100%, so that's good. It stays as is, and the rest of the tree stays as is. Okay. So now I have a new tree where I'm showing where the information is shown is 60% supported from the data. Okay. So you only have a tree that is highly supported by the data, and things that are not so supported is not really claimed by the tree. Okay. Make sense? Okay. All right. So an important distinction when you are looking at phylogenetic tree is the type of data that you use, and you could have species tree, such as when you look at the tree of life, you are looking at the tree of all different types of organisms. There's a species tree. Okay, it tells you how species are related to each other. Whereas the gene tree will give you how different genes are related to each other. Okay. So what makes this problem gene trees more complicated is that species can contain multiple copies of genes, and a gene may be missing from a species, so you can have multiples of a gene or zero numbers of the same gene. Okay, so that makes the gene tree more complicated to construct. All right, so in this example, what I'm showing is the sodium potassium ion pump proteins. And number one, you see number 1 and number 2. That means there was a gene duplication event. You had one gene in an ancestral organism, and the gene duplication event caused that organism to have 2 copies of the same gene. Okay. And those 2 copies will evolve separately. They will mutate separately. Eventually they will be different. They will have different gene sequences. Okay. If they are close enough, you would be able to tell that it was a gene duplication. But if they mutated too much, you may not be able to find out that they were a gene duplication event. Okay. So an easiest way to claim that 2 genes are a result of gene duplication is that they'll have high similarity and they are from the same organism. All right, so once you identify duplicated genes, let's say human 1 and human 2, then you need to consider those two genes separately. Okay. So human 1, when you try to find a homolog in another organism, you need to compare it with human 1. So that'll be homolog of human 1. And then human 2 may have its own homolog. All right. If a gene is lost, okay, let's say you had human 1 and human 2, and let's say we have Drosophila, which doesn't have a gene duplication event. Let me try to find this. Okay, Xenopus. We have Xenopus 1, but we don't have Xenopus 2. Okay. If you only look at one copy of the gene, so this one is lost. Xenopus currently does not have this gene. It was lost during the evolution. If you look at human 2 gene, okay, without looking at human 1 gene, and if you do a blast search, this Xenopus 1 gene will come up, all right, because their sequences will be similar. And that may make you claim that these two are sort of homologs to each other, which they are in a way, but you need to also look at other genes within the same organism that are highly similar and do the same search with those. And if human 1 is closer to Xenopus 1, then that's the true homolog that you need to consider. Okay? Human 1 is, then you can figure out that it's a duplicated gene whose corresponding copy in Xenopus is lost. Okay? So figuring out these gene duplications and gene losses is sort of a detective work. In some cases, it's clear-cut. In other cases, it's not so clear-cut. And there are different methods to help you identify these events. All right. So in this tree, the small squares represent a gene duplication. So there was a single copy of the gene, and then all the organisms, well, at this level, whatever that historical organism was, there was a gene duplication, and everything that evolved from that obtained 2 copies from that. And then those 2 copies evolved separately, and some of the organisms lost some of these copies, such as Xenopus 2 does not appear here. Catastomus 2 does not appear there. There was a secondary gene duplication event here. So human and chicken got 1 more copy. All right. Okay. So some terms here. An ortholog refers to so orthologous genes refer to genes that are similar to each other because a speciation event. Okay. So if you have human 1 and chicken 1, those 2 genes are orthologous. All right. Paralogs are due to gene duplication events. Human 1 and human 2 are paralogs. Okay. And I would say human 1 and chicken 2 are also paralogs. All right. Okay, onologs. Do you guys know what it is? I think it is whole genome duplication. So it's the same as paralog. You have a gene duplication, but instead of a single gene duplicating, the entire genome of the organism duplicates, and then you would still have more than 1 copy of a gene. But the reason you have more than 1 copy is whole genome duplication instead of a single gene duplication. Okay. How about xenolog? Yes, xenologs are due to horizontal gene transfer. That means you have 2 different species, a gene is transferred between them, and that usually happens in bacteria, where such as things like antibiotic resistance can be transferred from one bacteria to another. Okay, or you could have a viral genome being integrated into a species, then the genes from the viral genome would be considered xenologs. All right. So what type of data should we use for evolutionary reconstruction of species? There are two things we need to consider. First, the sequence should change, right? Because if it doesn't change, then we cannot tell the species apart. On the other hand, it should not change too much, because if things change too much, then we cannot really figure out that gene A is really related to gene B in a different organism. So we want to be able to find correspondences even at the gene level, both at the gene level. So we want to be able to identify homologous genes between two organisms, and also we want to be able to align them accurately. All right. When things change too much, you don't. The alignment that you produce from a sequence alignment algorithm may not reflect the evolutionary history of those residues. Okay. All right. So you need sort of an intermediate level of difference to construct the evolutionary history. Small ribosomal subunit RNAs have been found to be well suited for evolutionary reconstruction of species because they mutate fast but not too fast. Okay, based on the description that I just gave you. All right. So when we look at the evolutionary distance between species or between genes, there are some there are some quantifications that we need to do. The most common quantification is the p distance, which tells you the fraction of non-identical residues. Okay, so sequence identity refers to the fraction of identical residues in an alignment, whereas p distance is fraction of non-identical residues. Okay. P distance underestimates the number of mutations that actually occur. The reason is that if two things are different, okay, you don't really know, or let's say if two things are the same, you don't really know that they were the same in the first place, or they were different and then they both mutated and turned out to be the same. Okay. So cause of errors is that positions mutate several times, and since we don't have all the samples across the evolutionary history, we only have the leaf nodes. We don't know the exact number of mutations, and the rate of substitution is not the same for all bases at all locations. So when I described getting an ultrametric tree from an edited tree, I told you that we need to have an idea of a molecular clock that tells us how the rate of mutation relates to the evolutionary time. So the rate of mutation is described as the number of mutations that happen on the average, let's say, in 1,000 years. Okay, and the definition is at the very simple level. It says just a single number, but we know that some bases, some nucleotides mutate more frequently than others. Okay, one example is that when you have a codon, let's say AAA. All right. Hopefully that's a good example, but I'm just making it up. So usually the third codon can differ, but your codon will still give you the same amino acid. Let's look at the genetic code to find an example. All right, so let's skip that. Projection device is off. Okay. I'll wait. All right, so if we look at the genetic code, we see that the third position can usually vary. Okay, so let's look at here. The first two letters are CU. The third letter can be any of UCAG. They all encode the leucine amino acid. Okay, so what that means evolutionarily is that if you change the nucleotide at the third location of a codon, it does not change the amino acid for this case. For some of the other cases, it'll change. All right? So it has less functional consequence. Changing the third nucleotide has less functional consequence because in most cases, it doesn't change the amino acid being encoded. All right? And since it doesn't have any functional consequence, it will not have an evolutionary pressure. So if the protein is not changing, the function of the gene is not changing. So it doesn't really add or increase or decrease the fitness of the organism. So more or less, that mutation will appear randomly. Whereas if you make a mutation that will change the amino acid, it may change the fitness of the protein, and it will be selected for or against. Okay. So there's a different evolutionary pressure on the third base compared to the other bases of a codon. All right. So to handle these causes of error, we usually do a distance correction, and the distance correction tries to estimate the actual number of mutations that have occurred. So we can look at the p-distance and try to come up with a correct distance measure. So one observation is that transitions occur more frequently than transversions. So the blue is transition. Those are purines. So an A to G is more likely to happen than an A to T mutation. Okay. And if you look at a bunch of aligned sequences and count the number of mutations, you see that transversions happen less than the transitions. Okay, so observed mutations versus times its divergence, which is the distance between two species in terms of their evolutionary time. And as I just mentioned to you, coding sequence mutations have higher selective pressure. So if a mutation changes the amino acid, it'll have a higher selective pressure. If it doesn't change the amino acid, then it's sort of random. It'll appear at random. So in most cases, if it's a protein coding gene, it won't change, and it doesn't change the amino acid. It doesn't have higher selective pressure, but there are also other things before the translation, before the protein is produced, that may affect, let's say, how much the gene is transcribed or translated. Okay. So the expression level may depend on having a certain nucleotide. So we can still not rule out completely that there's absolutely no selective pressure. So when you mutate a nucleotide, even if it doesn't change the amino acid, it may change other things such as how much that gene is expressed, whether it is degraded by a certain, whether it's targeted by a certain microRNA and so on. Okay. Okay. So the definition synonymous is a mutation that does not change the amino acid, and non-synonymous is a mutation that does change the amino acid being encoded. The synonymous versus non-synonymous ratio can help us identify the type of selection. All right. A positive selection is where a mutation has caused the protein to function better. Okay. Whereas a negative selection means that the protein wants to stay as is. It doesn't want to change, so it's already optimal. Okay. If it's already optimal, you will have less non-synonymous protein, non-synonymous mutations compared to a positive selection. Okay. And And the synonymous mutation gives you sort of a baseline because it's more or less appearing at random. Okay, so the p-distance, as I mentioned, underestimates the actual evolutionary distance because of the errors such as the identical residues. You can have more than one mutation at a single location, and also you don't have the same mutation rate at all positions. Okay. The Poisson correction models the mutation rate, the mutations as a Poisson model. Okay. The probability of n mutations having occurred at time t is given by this formula. All right. The probability of no mutations is you just plug n equals 0, and you get this. Okay. Since you have two sequences, you want no mutations to occur for both sequences. So you multiply the two probabilities together, you get e to the minus 2RT. So 2RT is the distance that they are apart in evolution. Okay. So the average number of mutations is given as RT. Okay. So that is d over 2. The distance is, as I mentioned, it's the it's going to be 1 minus b. All right. And then you can solve for d if you'd like by taking the logarithm of both sides. It's ln1 minus b. So what this, in a nutshell, well, as a consequence, tells you that if you have the p-distance, if you know the p-distance, which is going to be a value here, the p-distance is the fraction of non-identical residues. Okay. You can plug it into this formula and find the actual distance. And it'll give you this new value right here. That will be the evolutionary distance between the two sequences. All right. So this takes into account that when two things are the same, it could be because there was a mutation that made them the same. Okay, so you are seeking the case where there were no mutations explicitly using a probabilistic model. All right. Okay, some of the other corrections, the gamma correction takes into account that mutation rates are different at different positions of the sequence. Jukes contour is a more widely used one, and it explicitly models the fraction of nucleotide conversions. And some of the other corrections involve distinguishing rates of transitions, transversions, because they happen at different rates, and also unequal base compositions. If you have more A's, then they are more likely to appear to be identical. Okay, so you try to account for things like that. Any questions? Okay. Is Daisy here? Okay, so I'm going to leave the floor to Daisy, who is going to go over some of the homework problems. And I'll come back and we'll implement a phylogenetic tree construction program.